In today's webinar, our objectives will be to give some background information on methylation and bisulfite conversion. We'll briefly touch on the Infinium methylation assay, but the bulk of the content will be discussing Genome Studio 2011.1, the methylation module. So we'll go through how to create projects, how to evaluate the built-in controls on the assay in Genome Studio, as well as another software package available from Illumina, the Beta-Array Controls Reporter. We'll then go through some data visualization tools in Genome Studio, and I'll leave you with some third-party analysis tools. All right, methylation is the chemical addition of DNA to which a methyl group is added to cytosine. This occurs in higher eukaryotes almost exclusively at CpG sites. So these are CG dinucleotides going from the 5' to 3' direction, and the cytosine can have a methyl group added. When this occurs, it typically happens on both strands at any given CpG site. So as you can see in the schematic at the bottom of the slide, we have a CpG site highlighted in the blue box, and you can see both strands, the cytosine has a methyl group added. Methylation can affect gene expression because the methylated cytosines end up compacting the chromatin structure, which can impede transcription factor access and silence genes. Tumors, in particular, exhibit abnormal methylation status and thereby gene expression. Methylation is a compelling target to study because the methylation state is dynamic and it can change in response to the environment, therapeutics, cancer, and other factors. One of the tools that can be used to study methylation is the Infinium methylation assay. So this makes use of Illumina's Infinium assay, in which DNA is extracted and processed on a bead chip. However, for methylation analysis, we're also adding in a bisulfite conversion step upstream of the typical Infinium assay. Bisulfite conversion is a chemical process in which cytosines are converted to uracil. This only occurs if the cytosine is unmethylated. So if a methyl group has been added to the cytosine, it will act to protect the cytosine from the conversion process, and it will remain as a cytosine after the bisulfite conversion reaction. So through this mechanism, we're able to infer the original methylation state of a cytosine by determining whether or not the bisulfite conversion reaction changed it to uracil. Importantly, only single-stranded DNA can be converted in the bisulfite conversion reaction, so this has some processing implications that we'll get into shortly. Bisulfite conversion will convert all unmethylated cytosines in the DNA to uracil, which are then later converted to thymine later in the Infinium assay. So throughout the presentation, you may notice I'm talking about cytosines or Cs getting converted to Ts, but this is just a proxy for the bisulfite converted cytosine to uracil and then uracil to thymine. So you can think of it as a C to T transition. Bisulfite conversion results in the loss of complementarity between the two strands of the genome. This is because the cytosines are converted to uracils, and uracils only make 2 hydrogen bonds, whereas guanines want to make 3. So in this example, here we have a piece of DNA shown. The cytosines are highlighted in blue, and a CpG site with methylated cytosine is highlighted in yellow. After bisulfite conversion, those blue cytosines will be turned to uracils, and now they will no longer be complementary to the guanines in the other strand. The cytosine, as you can see, will remain as a cytosine if it was protected by the methyl group. Because of this, our probe design in the Infinium assay must be target strand specific, and it also assumes that complete bisulfite conversion has occurred. We recommend using only the following validated bisulfite conversion kits. These kits are not included with the Infinium methylation kits that you purchase. Rather, they must be supplied separately by the user. The tech bulletin linked below shows the table that's displayed here, and we recommend searching the Zymo Research catalogs by the catalog number rather than the product name, because there are many very similarly named bisulfite conversion kits available from Zymo. Additionally, we suggest performing the Illumina recommended protocol modifications. These can be found in the Infinium HD methylation assay reference guide. So these are modifications to the DIMO protocol for bisulfite conversion. And again, they're detailed in that Infinium methylation reference guide. Specifically for the incubation step, we recommend adding 16 cycles of 95-degree temperature spikes, because this helps ensure that the DNA remains in a denatured state throughout that incubation. Because remember, the bisulfite conversion reaction is only going to occur if the DNA is single-stranded. So it's important for getting good bisulfite conversion efficiency to make sure you're doing this modified protocol for the bisulfite conversion. All right, so now to touch on the Infinium methylation assay itself. As I mentioned, this Infinium assay workflow goes from amplification to imaging on the iScan. And this is common to all of our Infinium assays, both for genotyping and for methylation. The unique thing about the Infinium methylation assay is that it has this bisulfite conversion reaction upstream of the typical Infinium assay. For the purposes of time in today's webinar, I'm not going to go through every single one of these steps in the standard Infinium assay. However, we strongly encourage you to refer to this Infinium Best Practices webinar for the details of each step. Today, we'll just be focusing on the steps that are specific to the methylation assay, so really zeroing in on that bisulfite conversion step. If you're working with FFPE samples, which are formaldehyde fixed paraffin embedded samples, you'll need to also do the FFPE QC kit and Restore kit. These kits should be done before and after bisulfite conversion, respectively. The purpose of these kits is to check the quality of the FFPE DNA by a qPCR step that's in the QC kit, and then after bisulfite conversion, to restore the DNA using the Restore kit. Importantly, the Restore kit should be used only after bisulfite conversion. It includes some amplification steps that would erase the endogenous methylation state. So it's important that that's done after bisulfite conversion. If you are working with FFPE DNA, please refer to the tech bulletin linked here. There is not a separate protocol guide for the FFPE samples. You'll just be using the standard Infinium HD methylation assay reference guide. But this tech bulletin supplements that protocol because it gives you specific volume guidance that differs from fresh or frozen cells. So if you're using FFPE samples, please refer to this tech bulletin for details. In general, the sample input requirements for the assay are that you need at least a minimum of 250 nanograms of DNA. But if you do have more DNA, we do suggest going ahead and using that, so long as you're using the same amount of DNA for all the samples in the project. You'll note in this table that more DNA is needed for the automated protocol. This would be processed with a liquid handling robot, such as a TCAN. The reason for that is just that there's more dead volume in the wells for those robots to pipette, so we can't end up adding all of the sample onto the bead chip because some is left in the wells. So if you do have a very limited amount of DNA, we suggest using the manual protocol. If you are using FFPE samples or degraded samples, again, the Infinium FFPE QC and Restore kits are recommended. Specifically, Illumina's validated kit is recommended because not all FFPE Restore kits on the market are appropriate for use with bisulfite converted DNA. Again, this DNA is then single-stranded, so that can have implications for some of the mechanisms that these Restore kits are using. The protocols can be found linked below. Now I want to get into the meat of our webinar today, which is discussing Genome Studio 2011.1 and how it can be used to analyze methylation data. The software is available to download for free on the Illumina website. You'll just need to make sure you have admin permissions on a Windows PC computer in order to install it. You can download it from the link provided here. Note that Genome Studio 2011.1 is the only version of Genome Studio that has the methylation module that's required. If you already have Genome Studio 2.0, that won't work for methylation data, but you can install both versions of Genome Studio on the same computer. That's not a concern. But we do recommend downloading 2011.1 if you're working with methylation data. So once you download it from this link, you'll need to extract or decompress the downloaded folder. and that will give you a folder that contains two documents. One is the license keys, and the other is the installer package. I know it's tempting to just immediately launch the installer package, but we really do need to open the license key for the first time you're installing the software. So in this license key document, you'll find a serial number provided for the methylation module. Simply copy that to the clipboard of your computer, and then you can open that installer package, check the methylation module box, and paste the serial number into the box next to that methylation module. Once you do that, you're ready to select Install, and Genome Studio will be installed on your computer. To create your first methylation analysis project, there are several raw ingredients that Genome Studio requires. The first is the .bpm manifest, which the software refers to as the content descriptor. So whenever you see it say content descriptor in Genome Studio, know that that's referring to the .bpm manifest. This is a file that includes information about all of the probes on the array, and it can be downloaded, again, from our website, illumina.com. If you simply type in the name of your B chip and then the word support, you can find the support page for that B chip and then go to the product files section to download the manifest file. For example, if you're working with Methylation Epic, you could type in Methylation Epic B chip support on our website search bar, and that would pull up the support page for you to download the manifest file. Genome Studio also requires a scan folder, which I'll talk about more on the next slide, but this is the folder that's generated by your iScan or your NextSeq when you're done scanning a B chip. You can optionally provide the software with a sample sheet if you want to provide specific sample names or provide certain grouping information about your data. Looking at an individual scan folder, so you need an SDF file and then red and green IDAPs for each sample. The SDF file is specific to each B chip, not each sample. But the scanner will write a folder that is named with the barcode of the B chip that was scanned. And then within that, you need the SDF, that's the Centrix definition file. That just contains some basic information about the array that the software uses to make the project. And then you'll need a red and green IDAP for each sample that was scanned. These are the intensity data files, and that's the raw data that is used in Genome Studio. So next, I'm going to go through a live demonstration of the clicks that are required to make a methylation project in Genome Studio 2011.1. Once you launch the software, you'll see this start page. You can either go to File, New Project, or under the New Project section in the center of the screenshot here, you can select methylation to launch the methylation creation, the project creation wizard. For the first step, it'll ask you which methylation assay you'd like to use. For that, you're going to select Infinium, and then you can click Next. Then I'll ask you more specifically which assay type. We want Infinium HD, click Next. It'll ask you where you want to save the project, what you want to name it, give it a name and click Next, and then where is the data saved on your computer. So you can browse to the folder that contains those scan folders, that parent folder. Click OK, and then from the barcode list, click the first one, hold Shift, click the last one, and then click the triple arrow button to add all the samples on those B chips to your project. You can click Next, and then it'll launch the group set analysis. So you can give it any name that you'd like to. And then we recommend selecting all samples again and clicking the triple arrow button to make a separate group for each sample. This helps make the project faster. And then for the analysis types, you can always change these later. There's different options provided. You can choose to subtract background or not. But for now, just to create the project, we're going to click Default. And then the content descriptor, remember that's the manifest, click Browse, and then browse to the .bpm manifest file on your computer. Only after you've selected that BPM manifest file should you click Finish. It might give you the option to click Next earlier before you've selected the manifest file, but we don't recommend doing so. You'll need to make sure you browse to that manifest before you click Finish. Okay, so now going back to these normalization options. I know I went through it kind of quick there in the demonstration for how to generate the project, but you can always go back and modify these later. So that's what we're going to do now. We're going to revisit these options. They're described in more detail in the Genome Studio Methylation Module User Guide that's linked here. It has descriptions of how these different normalization options work. If you're using third-party software for downstream analysis, most of these software packages include their own normalization algorithms, so you might choose not to normalize in Genome Studio if you're just using it to look at the controls. However, if you're only using Genome Studio to do all your data analysis, we do suggest normalizing your data. So again, from that parameters dropdown, you could select the controls normalization, and you can choose whether or not you'd also like to track the background. If at a later date you want to rerun a different analysis and change the normalization settings you've made, you can go to Analysis, Run Methylation Analysis, and you'll just need to give the analysis a new name, and then you can change whatever parameters you'd like. So this is what the workspace looks like once you've created a Genome Studio project. You can see the samples table on the left there lists each individual sample that's in the project. This demo data project is very small. There's only 3 samples, so that's less than a full B-chip. And then on the right-hand side, you can see the group methylation profile and the sample methylation profile table. These contain information about each CpG locus or each probe that's on the entire array. So you can see I've highlighted in red the row count there. So that's indicating how much content is on this array, and that's read in from that BPM manifest file. So this is the number of sites that are being assayed on the array. And I bring this up because it relates back to the B-chip specifications for the assay. So for methylation epic, for example, if you refer to the data sheet, there's a table here that shows what our specifications are for the number of sites that are detected. So using the standard protocol, that's fresh or frozen DNA, where our specification is 96% of the sites detected. And for FFPE samples or degraded samples, it's greater than 90%. And this only applies to non-cancer cells. So if you're accustomed to our Infinium assays and using our genotyping Genome Studio 2.0, you might be used to seeing the call rate specification expressed as a percent. However, in Genome Studio 2011.1, it doesn't do the math for you. So even though our specification is 96%, it's just going to tell you the absolute number of detected CpGs, and you'll need to do the math. So what we suggest doing is just remembering what the 96% or 90% specifications end up being. So for 865,000 samples or sites on the methylation epic B-chip, this is evaluated 96%, 831K, and for FFP degraded sites, the samples, the same number of sites, 90% works out to 779,000. So if you keep those numbers in your head, 831K or 779K, you can then look in the samples table at the detected CpG and see whether or not your project is meeting the specifications. So is it greater than 831K, for example, if you're using fresh or frozen samples? So the detected CpG metric is simply a measure of how many loci were detected over background. It's in no way reflecting what the methylation state is, and it's not telling us how well the bisulfite conversion went. So it's by no means the end-all be-all of your data QC, but it's a quick and easy place to start when you first open your project as something to check. So what we're really interested in Genome Studio is getting at what the methylation state was for each of these CpG loci, right? And since the readout from the B-chip is fluorescent signal, Genome Studio is using the intensity for the methylated signal and the intensity for the unmethylated signal in order to determine what is the beta value, which is representing our methylation. So signal A in the sample methylation profile table, for each sample, you'll have signal A in signal B. Signal A is the intensity for the unmethylated signal, and signal B is the intensity for the methylated signal. So these two signal intensities are used to calculate the average beta value for each CpG, for each sample. So the beta value formula is signal B divided by the total, so A plus B, plus a small correction factor. So here's that formula again. And if we're going through what the theoretical beta values would be for a methylated CpG, you can imagine that your signal B would be very high, right? That's your methylated signal from the B-chip. You'd have high signal B divided by a very low signal A, unmethylated signal, plus that same signal B. So your fraction would be a high number divided by pretty much that same high number. So that fraction would simplify to about 1 as your beta value. So for methylated CpGs, our beta values are going to be pretty close to 1. For unmethylated CpGs, we would have a low signal B intensity and a high signal A intensity. And so that fraction would be 0, or a very low number, divided by a big number, and that fraction would simplify down to beta equals about 0. So for unmethylated CpGs, our beta value should be close to 0. Beta values are intended for comparing the same CpG site between different samples or different groups of samples. It's not really intended for use within a sample comparing different CpG loci. So now that we have an understanding of beta values and how the methylation status is being calculated by Genome Studio, I want to go through how to check the data quality by evaluating the controls in Genome Studio. So Genome Studio 2011.1 has a controls dashboard, which can be accessed by going to Analysis, View Controls Dashboard. When you launch this, any samples that are currently selected in the samples table will be highlighted as yellow dots, which actually makes them a little bit difficult to see against the white background. So before launching the controls dashboard, we suggest clicking this double check mark button under the samples table twice, once to select all samples and then again to deselect all samples. That way you won't have any samples that are just showing up as yellow dots that make them really hard to see. I'm not going to go through the entire controls dashboard with you in this webinar for the sake of time. Instead, we're going to zero in on the bisulfite conversion and methylation specific controls and spend much more time on those. So for a full walkthrough of how to go through the controls dashboard for our Infinium assays, please see this recorded webinar. It is about genotyping samples, but again, all of the controls for the controls dashboard, many of them are the same between our genotyping and methylation B chips. So it's a very useful webinar. In the Infinium HD methylation assay reference guide in the appendix, there's a controls table that details all of the controls included on the array and what channel they should be evaluated in, whether it should be the green channel or the red channel, and what the expected intensity is for that channel. So these are the sample independent controls. And again, I'm not going to go through these in today's webinar, but please refer to that recorded webinar linked below for sample independent controls. This is staining, extension, hybridization, and target removal. Today we'll be focusing on the sample dependent controls. So our bisulfite conversion, specificity, non-polymorphic, and negative controls. So again, the same table has which channel they should be evaluated in and what the expected intensities are. We're going to start by talking about bisulfite conversion efficiency. And before we get into the controls, I wanted to give some background information about why this is so important to check on your B chip. So the methylation Infinium assay is assuming that all of the cytosines present after bisulfite conversion had been protected by methyl groups, and that's why they remained as cytosines. Incomplete bisulfite conversion before the Infinium assay can result in false positive methylation calls because we would have some unprotected, unmethylated cytosines that might remain as cytosines even after the chemical conversion if it doesn't go to completion. This is also problematic because incomplete conversion can decrease probe binding efficiency between the sample and the array because the probes are designed to the completely converted genomic sequence. This table is showing what we would expect for complete versus incomplete bisulfite conversion for unmethylated and methylated CpG loci. So you can imagine for an unmethylated cytosine, if we get complete conversion, all of those Cs should turn to Ts. However, if we have incomplete conversion, the cytosines might remain as cytosines, and therefore we'd no longer be able to distinguish them from methylated cytosines that were protected due to the presence of that methyl group. So these built-in bisulfite conversion controls are important because they test for the presence of any unconverted DNA after the bisulfite conversion reaction. There's actually two types of bisulfite conversion probes on the assay. These controls are for each type of Infinium probe. So I'm going to describe that in a bit more detail coming up. But all of these bisulfite conversion control probes are designed to target non-CpG sites in the genome. So their performance is completely independent of methylation, and it's only looking to test the bisulfite conversion reaction performance. So bisulfite conversion 1 is the control that's appropriate for our Infinium-1 type of probes, and bisulfite conversion 2 is to check our Infinium-2 types of probes. So what do I mean by these two types of probes? There's a tech note linked here that goes into this in great detail, where I've lifted this figure from. I highly recommend checking it out if you're not familiar with the different types of Infinium probes. First, I'm going to talk about Infinium-1 probes. So these probes have two different bead types for each CpG locus that's on the bead chip. One is designed towards an unmethylated CpG, and the other is for the methylated locus. For the unmethylated bead type, shown in the top left here, the probe ends in Ca, which is complementary to a CpG site that has been converted in the bisulfite converted DNA. So that's the green 5' to 3' DNA in the diagram there. You can see it's been converted. So the Cg, now 5' to 3', is a Tg. So that will be complementary to the Ca of the probe. When you do the single base pair extension reaction, a nucleotide will be added, just 5' of the CpG site in that bisulfite converted DNA, so to the 3' end of the probe. And the color of that probe will be dependent on what the identity of the base is in the genomic DNA at that location. So the cognate probe pair for this unmethylated bead type, now if you look at the bottom right corner, is the methylated bead type. This probe ends in Cg, which is going to bind to the GC of the bisulfite converted DNA if it was methylated and protected. So if it's not been converted to a T, we'll still have a cytosine at that location. So the terminal G on that methylated bead type probe will be complementary, and then we'll get binding and single base extension off of that probe if the site had been methylated and protected. So for each CpG site for Infinium-1 probes, we actually have two different bead types that are being analyzed, and we're looking for which of these two bead types we're getting signal from. And that is telling us if we're getting methylated signal or unmethylated signal. The bisulfite conversion 1 probes are similarly designed based on this Infinium-1 probe type design. So we have two different beads for bisulfite conversion 1, the converted sequence and the unconverted sequence. So just like the Infinium-1 probes I was talking about, the converted sequence bisulfite conversion 1 control probe will end in A, which will bind to the T in a bisulfite converted DNA, and the unconverted probe sequence will end in G, which would bind to any unconverted DNA. Remember, all of these bisulfite conversion probes for the controls are targeting non-CpG sites. So we're expecting to see complete conversion because there's no reason for that cytosine to have been protected. So if we're looking at the controls dashboard, these graphs shown at the bottom for bisulfite conversion 1 green, if you look at the figure legend, you can see that they're labeled C1, C2, and U1 and U2. These are the probes that are evaluated in the green channel here on the left, and we're expecting to see high intensity from the converted probes and low intensity from the unconverted probes. So these, again, are probe pairs with C1 and U1 being a pair and C2 and U2 being a pair. What we want to see is that the green probes have higher intensity than the blue probes in that figure legend. Similarly, for bisulfite conversion 1 red, we're looking at whether the purple, reds, and oranges are higher than these like orangey yellows. So whether the C's are higher than the U's. And what we want to see is a good gap in intensity between the converted and unconverted signal in each channel. Now turning to Infinium-2 probes. So other CpG loci on the bead chip have probes that are designed using the Infinium-2 probe type. In this assay, there's only a single bead type that will report both the methylated and unmethylated status using color. So these probes end in C, and then the single nucleotide extension reaction will fill in what base is complementary to that critical CpG, that cytosine that either has or hasn't been converted. So from which nucleotide gets added from single base extension, we can determine whether or not the bisulfite converted DNA contained a T and had been converted, and thus was unmethylated, or whether it still has a C and had been protected and thus was methylated. So in the Infinium assay, any A's that are added end up being stained with red antibodies and they'll show up in the red channel, and any G's that are added are stained green and will show up in the green channel. So in Infinium 2 probes, the color is indicating the methylation state. So looking at the bisulfite conversion 2 probes, these controls are again evaluating whether or not we had good bisulfite conversion. So what we're hoping to see is that all of our DNA has had a T at that bisulfite converted DNA site that's being assayed by the probe. So when we get our single base extension and then staining, we're hoping that we'll see red signal, because that would indicate that there was a T present in the bisulfite converted DNA, and therefore that cytosine has been converted to a T. So we're evaluating this actually in both channels because we want to see low or no signal in the green channel. That would be any remaining unconverted cytosines. And we want to see high positive signal in that red channel because those are our converted cytosines that are now being read as thymine. So you just need to check for a signal in the red channel and no signal in the green channel for our bisulfite conversion 2. Additional sample-dependent controls for the methylation assay are specificity. These are also divided into Infinium 1 and Infinium 2 probe types. Specificity 1 is for the Infinium 1 probes, and here we're really looking at the specificity of that single nucleotide extension of the Infinium assay. So we have a perfect match bead where we have the probe that ends in A, which we're expecting to see extension for the genomic region that has a T. And if the probe instead has a mismatch and ends in a G, we would not expect to see any extension off of that because we'd have a mismatch between the G and the T. So looking at the controls dashboard now, the specificity 1 green, we're again looking to see are perfect matches higher in intensity than our mismatches? Are the greens in that figure legend higher than the blues? And similarly for the red channel. So we want the purple, red, and oranges to be higher than the yellows because this is a higher intensity from our perfect matches than our mismatches. Similarly for specificity 2, these are now Infinium 2 probes, and this is targeting a non-polymorphic T within the genome. So we should only be seeing extension and staining of an A, right, that's added to the probe. So we want to only see signal in the red channel here, not in the green. Lastly, the negative controls. There's about 600 different negative control probes included on the bead chip, and these are not targeting any site within the genome at all. So we're not expecting to see signal from them. We're hoping to see low signal in both channels. And what these probes are showing, or what the dots are showing, is the average and standard deviation for all 600 of those probes for each sample. All right, well, that concludes how to look at the controls dashboard in Genome Studio. Another option for evaluating these controls is using a more numeric approach in the Bead Array Controls Reporter software. So this is a high throughput quality control software that's specific for Infinium methylation data. It's very fast and easy to use, and again, it's formula derived, so it's using numbers to evaluate the controls instead of visually looking at those plots and evaluating the relative intensities. It can automate the analysis of the control probes and will compare the relative intensities. You know, is that green higher than the blue? It's doing that automatically for you and flagging unexpected results. This is intended to be a companion tool for Genome Studio, not a replacement. The Bead Array Controls Reporter will visually flag samples that might need further evaluation in Genome Studio, but Genome Studio is still useful for sorting and identifying any trends or patterns in the data, as far as if it's a particular row or particular column of your plate that was affected and so forth. So Genome Studio is still very useful for troubleshooting. Genome Studio also provides the detected CPGs that we talked about, as well as the intensities, right, that signal A, signal B, and other data analysis options. But Bead Array Controls Reporter is only looking at the controls. There's a webinar here that has more details on the Bead Array Control Reporter, but for right now, we're just going to skip to how to use it and what the report looks like, so you can quickly decide whether this is something you'd want to include in your analysis pipeline. To install Bead Array Controls Reporter, you simply need to download the software from our website. Again, it just requires Windows. And then the second link here is the software guide, which contains helpful formulas that are actually what the software is doing in order to derive the values that are shown in the reports. This is on page 9 of the software guide. It also has helpful information about the probe design for the controls and how they're interpreted. So even if you don't end up using Bead Array Controls Reporter, I've heard from some people that it really helps them get a deeper understanding of the controls for the methylation data. So once you download the installer, the software will look like this orange eye icon. And if you open it up, you'll see the Bead Array Controls Reporter. It's very simple. It simply has a IDAT file location you browse to and a start button. I'll go through a little bit more of the options here for how to generate a report. The first step is to set the output directory, which is where the report will be written. So this is where it will save the report that's generated. If you don't bother to specify a location, it will just save it to your documents folder. Then the only other thing you need to do is tell it the IDAT file location. So this is a parent folder that needs to contain all of the following files. It needs your IDATs. So these can be housed in scan folders. You can just leave them how they come out of the scanner. But you also need to put the sample sheet saved as a .csv file and the manifest in BPM format in that same folder. You can see in the screenshot here on the right that all of these files, the IDATs, the sample sheet, and the manifest, they're all in a folder that's called Demo Data Epic. So that Demo Data Epic would be the folder that I browse to as my IDAT file location when I'm using Bead Array Controls Reporter. If you need a sample sheet template, that can be found on our website at the link here. Also, along with those BPM manifest files and some demo data, which has IDATs if you don't have any data yet, but you want to practice and see what the Bead Array Controls Reporter looks like. So to start the analysis, you just need to browse to that IDAT file location that contains all that data and click Start Analysis. The report that's generated will look like this. It's a spreadsheet, so it's .csv format. The sample name and bead chip information on the left side in columns A, B, and C are pulled from the sample sheet that you provide. And then going across each of those column headers are the names of controls from the controls dashboard. And so what we have for each sample, each row of this spreadsheet, we have a value for each control. And what the Bead Array Controls Reporter has done is it's used the manifest file you provide to look in those intensity data files, those IDATs, for the red and green intensity for each control probe. And then it's applied the formulas outlined in the user guide in order to derive the numbers that are shown here. And then the last step is that it uses a threshold to decide whether or not to flag it. So you can see there's some values that are highlighted in yellow here. These are samples that failed to meet the threshold. In this case, for separation between the converted and unconverted probe intensities for the bisulfite conversion controls. So you can see how this would immediately stand out if you're evaluating a report from a large project. Anything that's highlighted in yellow, you might want to look in Genome Studio and see what's going on with those samples. You can change the thresholds. If you're using the FFP Restore Kit, you'll want to change the restoration threshold from 0 to 1. In order to launch the window to set the thresholds, you need to go to Options, Set Thresholds, and it will open up this the screen that's shown on the right here. We recommend that the values that have a default of 1 should not be modified. There's more information on this in the user guide. However, the ones that have a default value of 5, you can tailor if you'd like to make them more stringent for your setup. The very bottom value here, the background correction offset, this is a global setting that's used for the background value, and Illumina has empirically derived the value of 3000 to be used as default, and that does work for most users. However, if you're finding that Bead Array Controls Reporter is flagging like all of your data all the time, even when it looks good, you might need to change that background correction offset because your scanners might have a different laser power. You can give us a call in tech support, and we're happy to help with that. So that concludes the ways of looking at the controls, both in Bead Array Controls Reporter and Genome Studio. Again, it's always using the same control probes for both methods, but the values are being automatically compared to the background that's appropriate in Bead Array Controls Reporter. and highlighting any things that need to be looked at in more detail. Another advantage of Beta Ray Controls Reporter is that it can analyze very poor quality samples that might cause Genome Studio to hiccup during project creation. So if you're ever having trouble with Genome Studio project, one thing you can do is try running the Beta Ray Controls Reporter and see if there's any samples that stand out as particularly problematic. All right, so the last main part of this webinar is to go through some data visualization tools in Genome Studio. One metric that we recommend checking is the detected CPGs across the project. If you have many samples in your project, rather than scrolling down the samples table to look at this, it might be easier to use the line plot. You can click this mountain-shaped button in the samples table toolbar. That's the line plot icon. And that will launch the line plot tool, and you can simply select detected CPG from the columns in order to plot the CPG for each sample. So this project only has 3 samples in it, but if you had many more samples, this can be used to quickly identify any outlier samples that you might want to look at more carefully. You can also choose to layer in a second line on the line plot. So if you click the right axis tab there, you can layer in either the signal average for the green channel or the average signal for the red channel. So that might also help you just look at your data in a different way. Another favorite plot is the box plot. This one can be used to plot the average beta values. So in the sample methylation profile, in the toolbar, click the box plot icon, and then you can select the average beta value. So this is an average of all of the CPG loci on the entire B chip for each sample. So each box plot is for one sample, and it's looking at the average for all of the beta values on the chip. This is also great for identifying outliers and seeing if there's any differences between your samples on a very high level. Next is the histogram plot. So in that same methylation profile, just one button over to the left is the histogram button. So with that, you can select a specific sample that you'd like to look at under the columns menu, and then select average beta value under the subcolumns. And you can see that this will generate a histogram with different bins for different beta values. We typically see a bimodal distribution with peaks around 0 and 1 corresponding to the methylation state. You can also analyze replicate samples in Genome Studio to determine what the R-squared is as a way to assess the reproducibility. So for this, if you've had any filters active, we first recommend clearing them by clicking the funnel button with the X. And then you can launch the scatter plot tool, which is just to the left of the histogram button. And with that, in the columns section of the pop-up menu, you can select the first sample you'd like to compare, and then select average beta for the subcolumns. Select which axis you'd like to apply the sample to, for example, y-axis, and then do the same thing for the second sample you're comparing. So select a different sample in the columns, select the average beta again, and then click the x-axis, and then click OK to build the scatter plot. So the scatter plot is interactive. There's actually a lot of different things you can do with it in Genome Studio, but for the purposes of right now, what I wanted to show you is at the very bottom, that R-squared value that's highlighted. So it's greater than 0.99 between replicate samples is what we're expecting. But you can highlight any CPG on this plot, and it will be highlighted in the sample methylation profile table. You can also use the endfold to highlight loci that are varying between your 2 samples above a certain endfold change. So it's a very cool tool. You might want to play around with it. Another favorite way of looking at methylation data is, of course, looking at heat maps of the beta values. This can be generated in Genome Studio, again in the sample methylation profile. Click the heat map button, and then with the average beta default selected, you just need to click the sample subcolumn value to apply the average beta values to the heat map, and then choose the desired row label. So for example, you can select index and then click row labels to apply that to the heat map, and click OK. And what you'll end up with is a heat map of the beta values for each sample for each CPG. So at the top, we have each sample, and then going down, we have all the CPG loci, and their beta value is shown by the color that's indicated in the figure legend. So in this case, a beta value of 0 is shown in green, and a beta value of 1 is shown in red. If you'd like, you can also apply clustering to this data by clicking the clustering button in the heat map tool. And you'll just need to give the cluster set a name and select the desired clustering metric. The default is Manhattan. These are just different error models, and you can read about the different options in the methylation module user guide if you want more information. But just for right now, you can select any one that you want to cluster it, and it will build a phylogenetic tree of this clustering dendrogram, and you can see which samples are more closely related based on their data values to one another. All right, so that concludes the visual data analysis tools in Genome Studio that we wanted to go over, and I wanted to leave you with some third-party analysis tool options instead of Genome Studio. So the first of these is Sesame. This is an R package for looking at Infinium data, specifically Infinium methylation data. The paper is shown here from Dr. Peter Laird's group, and this is an R package, as I said. So there's a user guide and a GitHub link provided. All of these third-party data analysis tools are supported by the authors who made them, not by Illumina, so they're third-party. So if you have any questions or issues with using them, please reach out to the software developers for assistance. So this is a really great tool that has lots of really advanced normalization options and data visualization tools. A great R package for analyzing Infinium data. Similarly, MinPhi was kind of the predecessor to Sesame. It's another R package that's been used by many methylation customers. The paper is shown here, and you can check out the R package at the link shown below. We also have some videos, and if you Google, you can find a whole lot more documentation about how to use MinPhi if you're not accustomed to using R. And the last third-party tool is the DKFZ. This is the German Cancer Research Foundation. They've made a tumor classifier that's used for central nervous system tumors based only off of raw Infinium methylation data. So you can upload your IDATs to this tool, and it will compare it to the database and provide you with a possible tumor classification. The documentation is shown here. And again, all of these third-party tools, you'll need to reach out to the developers if you have any questions about them. But we just wanted to include them so you're aware of what else is out there in case you want to go beyond what Genome Studio offers. With that, I leave you with some resources that you can refer back to later, and I'll take any questions.